Hey guys, so this week we are talking innate immunity. Let's get into it. We're just starting out talking about the immune system, right? The immune system is really complicated. We're going to be watching some videos this week and next week to kind of give us a better synopsis of what's actually going on because there's a lot. So usually how we typically learn the immune system is we start with two arms of the immune system, if you will. And so those are the innate immune system and the adaptive immune system. Sometimes the innate is called the natural immune system. That's really dumb. Nobody calls it that. So the innate immune system is your basic first line of defense. So that's usually your anatomical barriers, okay? We could also argue that the extended immune system, so that would be like avoiding people who are sick, or if you see someone with symptoms of being sick, like just noticing that and being like, ooh, I'm gonna just keep away from you. You're all snotty and gross and red and inflamed, so I'm gonna avoid you. So we're kind of ignoring that. That's less something that we talk about in a class like this, but we're talking more like you physically as a mammalian host is usually what we're talking about, and usually it's humans or mice is what we're almost always talking about. And so the anatomical barriers that we physically have, like our skin, every kind of surface that touches the outside world, including your gastrointestinal tract, those barriers are super important because if you get rid of those, you're gonna die. That's what usually kill. Well, there's two things that usually kill people who get like burns all over their body. They either get an infection because stuff can just get in and just infect those delicate cells underneath, or they actually get dehydrated. That's one of the biggest problems because you don't have your skin to keep hydrated. But there's also cells associated with the innate immune cells or innate immune system. And so that's your typical, people usually think of macrophages, but we're going to talk about other cells too, like granulocytes. That's like your neutrophils, your eosinophils, your basophils. And we've also got natural killer cells. We're going to talk about what those do. Those are very cool. We're not talking about the adaptive immune system this week. We're going to spend the next 2 weeks talking about the adaptive immune system. We're going to spend some time talking about antigen presentation. And then we're going to talk about the humoral immune system. So that's how we make antibodies. And then we're going to talk about T killer cells and how they actually work, which is really, really cool. And so you can see just this picture there's a lot going on, but I'm going to take you through it. It's going to be awesome. Let's get to it. Let's talk about those barriers, okay? And so the first barrier that obviously that we think of is skin. Now, I don't have a picture for every single one of these little things, but I did want to just kind of go through these kind of from the surface of your skin and going down, okay? But they're all kind of associated. So first off, the commensal bacteria. So when we say commensals, that's as opposed to like a parasite or a commensal or a beneficial relationship, right? And so commensals are supposed to be like nobody's benefiting, or the host is not benefiting or being damaged for something. Now, I would argue that I can always make a case that a commensal is actually a beneficial bacteria or whatever that's living there. Because the presence of a commensal bacteria on the surface of your skin is actually really important and can help keep, it will use up nutrients, it'll use up space that other pathogenic bacteria could possibly use. And so that's a really nice thing. Because it's in the commensal bacteria that live on you's best interest or live inside of you. It's in your best interest, in their best interest, to keep you alive. And so keeping bad guys away is a good idea. And so this was an interesting, I found all kinds of cool articles because I rebuilt this presentation from the ground up. And I found some really cool articles. And this is just one that talks about like different places on your body, like on your foot, if it's dry or wet, in the sebaceous glands, like what lives in there. It's kind of cool. Hey, look at that, acne, the bacteria responsible for certain acnes. Also, the intracellular junctions, intercellular junctions, I'm sorry. If you take cell biology, we talk about this, but generally we're thinking like the tight junctions, the desmosomes, the hemidesmosomes, adherens that keep your epithelial layer nice and tight. And that's really important because you don't want bacteria sneaking, in between your cells through that paracellular passage. And also, so up here we've got, it's showing intercellular lipids and just we would call this the acid mantle. And so that's all the salts and the sweat and the sebum and just all the stuff that sits on top of your skin that you wash away if you use soap, interestingly. So you don't want to wash your hands too much. That can actually make you sick. And so the acid mantle is slightly acidic. And so it can get down to like 5, but it tends to be like 5 or 6. But even just having something that acidic can really change how bacteria live. What else? Desquamation, so that would be like losing dead skin cells on the surface. So that's like this nice little keratinized layer up here of dead cells. You could lose those. and it'd be fine, right? So if there's a bunch of bacteria on one, you could just, have it slough right off, and then you're good. They fell off with it. Uh, Dehydration. Your skin in general on the surface is actually relatively dehydrated, and so bacteria don't like that. Now, something else is antimicrobial peptides, and so antimicrobial peptides or AMPs. There's so many of these, it's crazy. We could spend like an entire class just talking about each of these. And so they come in a variety of colors, a variety of shapes. They do all kinds of stuff. Defensins are usually the one that people talk about. There's obviously a bunch of other ones, but they attack different bacterial cell walls in different ways. That's generally how they work. For the most part, what they do is they kind of stick into the cell wall and can disrupt it enough so that they basically all the contents of the bacteria spill out. They can be relatively specific. So one of the things that gets talked about with the innate immune system is specificity. The adaptive immune system, your B cells and your T cells, is incredibly specific, like hardcore specific to. It's going to be nuts when we talk about how specific they actually are. Now, your innate immune system, sometimes people will say non-specific. I don't like that term because there is a lot of specificity that goes into this. I mean, your alpha defensins might not kill every bacteria that a beta defensin kills, and vice versa. And so you need a variety of molecules to actually do that. We see that as well with our recognition receptors, which we'll talk about later. Those are in innate immune cells, and they're very specific to specific pathogens and specific antigens on specific pathogens. Now, the other mucosa, so this is every kind of mucosa you can think of. So usually we think of like nasopharynx, your lungs, all of your gastrointestinal tract, any urethral tract, so it could be like in the penis or a female urethra, and the vaginal tract as well. And so those are all those external mucosa that they're still technically facing out, even though they're sort of like inside of us, but they still do face outward. So those mucosas can vary based on the actual type of mucins. So mucins are the proteins that are actually associated with mucus. Like, what is mucus? So that's a good question, and there's people that actually study that. So mucus is a bunch of goopy stuff, obviously. I mean, we've all sneezed. But what is it? Well, it's actually a lot of protein, more than you would think, and also a lot of lipids and sugars as well. But you can study specific mucins. So some of my background, when I was in grad school, we actually were working on a bacterial vaccine, right? And it was a lactobacillus. And I'm going to talk a little bit in a minute about that. But there's a mistake on here, or on this picture. It's not a mistake so much as a difference in what's actually true. So down here it says impenetrable to bacteria, and that's interesting. So in the intestinal tract, your mucosa has a variety of thicknesses of mucus, and there are two layers that have also different depths, and it depends on where you are. And so like in the small intestine, it's not as deep. In the large intestine, it tends to get deeper. But there are two layers, the outer layer and the inner layer. Lots of bacteria can tend to hang out in that outer layer. And that's one way that we get bacteria out of either your intestines or out of the lungs or the vaginal compartment. Like, that's just a great way to get those out. That inner mucus layer is really, really thick. Like, it's crazy dense. And it says here, impenetrable to bacteria. That's not entirely true. For the most part, it is. Your bacteria like to stay in the outer mucus layer and in the lumen, where we can get rid of them. But there are bacteria that can hang out in that inner mucus layer, and our vaccine bacteria being one of them. And trying to get it to actually pass through that mucus layer was really important. Other pathogens can also do that. And so that's how they get in there and, I don't know, give you an infection. Probably the most famous is Helicobacter pylori, which actually goes through the stomach lining and can go through the stomach acid lining through the mucus layer that protects your stomach lining from the acid of your stomach, and can actually go in there and form ulcers. And that's the bacteria that causes ulcers. So if you have like an ulcer in your stomach, it's from a bacteria. Having acidity, like I just said, like your stomach is super acidic, and it gets less acidic as you pass away from the stomach. But like the small intestine and the large intestine are slightly more acidic than than pH 7, I guess. And so that can help as well against bacteria. And so usually the bacteria that we actually do see living in these places are acid tolerant, They're bile tolerant. They're able to resist enzymes. There's tons of enzymes. I mean, think like you have to eat food and break it down. So if you want a bacteria to survive that, it has to be able to resist enzymatic activity. It has to be able to resist everything that we use to break down food. And so that's an important set of skills that a bacteria can have, especially if you're trying to make a vaccine. Now, the actual expulsion of the mucosa is really interesting. Some immune books will list like vomiting or diarrhea. I mean, those are technically part of your anatomical barrier expulsion. So yeah, I tend not to think vomiting as like an immune system thing, but I mean, I guess it is if you're that sick. Though usually we think about it more on a cellular level with the cilia beating. And so I think I have a picture here of in the lungs especially, and in the vaginal compartment. We see a lot of cilia that will slowly push the mucus layer out, and that mucus layer is really sticky and it'll hold on to stuff and it'll push things out, which is really, really nice, and it can keep bad guys out. So moving on from our anatomical barriers, and we want to talk now about our cells, okay? So this is a slide you're going to see a lot throughout this course. So this is one that I adapted from somewhere, and then I kind of modified it a little bit to talk about the development of immune cells. So that's called hematopoiesis. That would be starting with a multi-potential hematopoietic stem cell, which would come from an earlier stem cell. And then it's distinguished enough to be able to form just immune cells. But that's a lot of cells that it can become, but it can't become anything. And so we see that when we study stem cells, which we talk about stem cells a little bit in cell biology as well. And so if you take a multipotent hematopoietic, that can become pretty much anything. We've got our myeloid progenitors and our lymphoid progenitors. That's the two big arms that they can become. And so your myeloid progenitors are generally going to become what we consider innate immune cells. So that's your granulocytes and your antigen-presenting cells like macrophages and dendritic cells. Now, we lump of our lymphoid progenitors, most of them are part of your adaptive immune system. So that's your T and B cells. Plasma cells being like the end B cell that actually makes antibodies. So, I mean, I would still call it a B cell, but hey, we call it a plasma cell sometimes. But natural killer cells are lymphoid progenitors, but they're generally considered innate immune cells. We're going to talk about how they actually work, so don't worry too much. Now, I wanted to cut to a crash course. So if you've watched any crash courses, they're awesome. They're really good. So this is a crash course in human biology, and the last 3 lectures of that actually talk about the immune system. And so I want to cut to a small piece of that video that really introduces the idea of the innate immune cells and their jobs. And then we're going to go into them specifically and what they do.
So when you've breached that first simple line of defense, it's time to call on your second line of internal innate defenses. This is where your body starts pulling strategic maneuvers like firing up a fever, releasing chemical signals, causing inflammation, or other defensive tactics that help identify and attack infectious invaders. Some of the first defensive cells on the scene are your phagocytes. Their name literally means to eat, and like Pac-Man, they indiscriminately chase down intruders and gobble them up. And they come in a few different varieties. First, you got neutrophils, which are the most abundant type of your white blood cells. They kind of self-destruct after devouring a pathogen, and in fact, you've probably seen piles of their little dead bodies, because that's what pus is made of. But the bigger, tougher phagocytes are the macrophages. They're derived from monocyte white blood cells that have moved out of the bloodstream to occupy tissues, and some are free types that patrol tissues looking for creepers, while others are fixed, attached to fibers in specific organs, devouring anything suspicious that passes by. So when a macrophage, in say the finger I just cut slicing a bagel, sees a new bacterium coming along, it snares it using cytoplasmic extensions, reels it in, completely engulfs it, and essentially digests it and spits the rest out. And unlike neutrophils, it can do this over and over again, like a boss. But not all your defense cells are phagocytic. You've also got cells with what is by far the awesomest name of any cell in the body, the natural killer cells. You can call them NK cells if you want to, but like, why would you do that? Anyway, these tiny assassins patrol your blood and lymph looking for abnormal cells, and are unique in that they can kill your own cells if they are infected with viruses or have become cancerous. How can they tell? A normal, healthy human cell contains a special protein on its surface called MHC1, or Major Histocompatibility Complex. But if it's infected, it stops making that protein. And if an NK cell detects a defective cell, it doesn't swallow it whole like a macrophage. It pokes it with an enzyme that triggers apoptosis, or programmed cell death, which is pretty awesome.
All right. That was Crash Course talking about specific innate immune cells. We're going to watch some more of their videos, as well as a Kirogestadt video later on that talks about the adaptive immune system and the connection between those two things. That's antigen presentation. And so let's start going. We're just going to go through most of the cells that they mentioned, a couple that they didn't. And so we're going to start with, if I go back here, so we're going to start with our granulocytes, okay? Why are they called granulocytes? Well, they have granules in them. So if you look at them under a microscope, they actually have these like little balls of stuff, and those are called granules. And you can stain them with different colors, and depending on the color that your granules actually stain is where we actually get some of the names for these. Now, neutrophils are the most basic defensive immune cell. They are the most common leukocyte. They have a bunch of different functions. I liked this one. I just thought this was a cool picture that shows how they move in the bloodstream and then come through there. That's called extravasation, and we are going to talk about that later on in the course. But I did like this picture. I thought it was cool. They do a lot of different things. Mainly they degranulate. That's generally what we think about when we think about neutrophils. So they throw their little bodies and they explode and they shoot out granules and it kills stuff, pokes holes in things. They can also engage in phagocytosis, and I have a video I'm going to show over this, of a neutrophil phagocytosing, which is generally something we don't think about with neutrophils, at least in modern immunology classes. But actually early on, when we first discovered neutrophils, their original name was microphage instead of macrophage, because we saw these two types of cells. There were really big ones that ate stuff, and there were really little ones that also ate stuff, i.e. bacteria. And so we called them microphages. But now we call those neutrophils because we saw that they stain with neutral colors, and so that's where the phile or the phil comes from. So neutral staining lover is what the name comes from. The other reason I picked this picture here is it's showing exocytosis, i.e. reactive oxygen species and degranulation, but it's also showing netosis. So netosis was something that was not discovered until like the mid-2000s-ish. Net stands for neutrophil, because that's what the cell is. Extracellular, okay, because it's outside the cell, so you can see all this stuff. Trap, okay, so it's a trap. And so what the neutrophil is doing is it's taking all the contents of its nucleus. So you can see this was the nucleus. They have very distinct looking nuclei. This is really what they look like under a microscope. And it's vomiting out all of the DNA and proteins and histones and all the crap that's inside the nucleus out into the extracellular or outside of itself, so the extracellular matrix outside of itself, and it traps bacteria. So you can see they got like, these look like Helicobacter pylori to me. All these bacteria with these flagellated bacteria, and they're trapped in there because DNA is really sticky.
In this scanning electron micrograph, neutrophils are attached to a flat surface, and we can see the abundant folds of their cell membranes. We can also see that the neutrophils are infected with the gram-negative bacteria Shigella, which appear as rod-shaped objects and are colored red. Also visible, in blue, is a fine mesh of fibrillar material called a neutrophil extracellular trap, or net, that is able to capture many of the Shigella bacteria. A net is created when a dying neutrophil extrudes the contents of its nuclei, including chromatin, into the surrounding environment through a special form of cell death called netosis. In this video, human neutrophils were stained with a dye that turns their nuclei blue, and then chemically activated to induce netosis. The media used to grow the cells contains an antibody that fluoresces green when chromatin is extruded from the cell, which allows us to view the process that creates nets. As shown by the timestamp, the video compresses 4 hours of real time into approximately 13 seconds. The nuclei of healthy neutrophils typically have several lobes, as we see here at the start of the experiment. However, after approximately 30 minutes, the nucleus in this cell, as well as many other cells, begins to disintegrate, as we can infer from the breakdown of the lobular pattern and the diffusion of blue dye throughout the cytoplasm. After an additional 15 minutes, the cell extrudes its contents into the extracellular space, as shown by the green clouds. As the experiment continues, additional neutrophils undergo netosis until about half have generated extracellular nets. During infection, microorganisms become entrapped in the nets, which makes it easier for neutrophils or macrophages to phagocytose the intruders. Patients with chronic granulomatous disease have trouble forming reactive oxygen species, or ROS, a necessary ingredient for the formation of nets. Subsequently, these individuals experience reduced net formation, and it is believed that this may contribute to their susceptibility to microorganisms.
Really cool. Something else that neutrophils can do is present antigen. That is definitely not their main job, but it is a job that they do. And that's something that's happened probably within the last 10 years, as we've been finding that a lot of cells that we didn't consider antigen-presenting cells actually do present antigen. Which is really interesting, but it can distract from actually learning how antigen presentation works, because your main antigen presenters are your macrophages and your dendritic cells. So let's actually get to those after we cover the rest of our granulocytes. So moving on to our next granulocyte is our eosinophil. So they're relatively uncommon. If you do a complete blood count and you have a really high eosinophil count, if you live in the Western world, if you will, so the United States or Europe, then you tend to, if you have high eosinophils, then they will say that you probably have an allergic reaction to something. Because eosinophils get upregulated when you have the presence of allergic antigens. What's really interesting, and something we'll talk about a little bit later in this class, is parasites and allergens, like ragweed or peanut butter or whatever, how we react to those two things, even though they seem very different. There are a lot of similarities in the antigen structure of parasites and allergens. It's really, really interesting. And so what we actually tend to see is that parasites will put surface antigens on themselves, okay? Because parasites tend to be really big. So we're talking big eukaryotes like malaria or schistosomiasis, or I don't know, you name it. And it will put antigens on their surface that have a lot of similarities to allergens, a lot of proteases that will act as antigens, and so that can attract a lot of cells. Eosinophils being your main granulocyte that we're going to see when we see either a parasitic infection or an allergic infection. So I said, like if you live in the Western world, then if you have high eosinophil counts, then you probably have an allergic reaction, unless they do a history and you just visited, I don't know, Brazil or sub-Saharan Africa or somewhere where parasites are a little more endemic. And in that case, they may think that you have some kind of parasite. So like malaria would probably be what I would go to. And so because you'd have high eosinophil counts. Now you can see, I grabbed a lot of pictures for these that just show, oh man, all these cells do like a million things. For the most part, eosinophils, and I'm going to cut to a video right now, eosinophils will swarm a parasite and degranulate. And degranulation, i.e. releasing bombs of weapons that just explode and kill stuff, and they're awesome. Okay, and so this video is really cool because it shows like a little worm getting killed, and so that's generally what eosinophils do. Now they also can degranulate and force mast cells and basophils to degranulate and release histamine, which is going to cause an allergic reaction. And so it's a really tricky balance that you have to strike when you're dealing with allergens versus parasites. So let's talk about basophils and mast cells. Now, they're also antiparasitic. Basophils and mast cells used to get really lumped together all the time, and in a lot of immunology texts, they still do, because most people don't actually study basophils or mast cells, even though they can cause a lot of problems, especially mast cells. So those are the cells that if they degranulate and they release histamine, then that can cause a really severe allergic reaction. Basophils can do the exact same thing. Now, in general, they're like eosinophils. They're also associated with antiparasitic properties, and so they're good at degranulating in the presence of parasites. They also tend to stimulate a T helper 2 response. We'll talk about that difference later on. T helper 1 would be like how a T helper cell that wants to destroy a virus infected cell, and you use T killer cells to do that. So a T helper 1 response tends to be a T killer cell response. It's what the T helper cells are driving. A T helper 2 response tends to be what a T helper cell would do if it wants to drive an antibody response. And in general, you want to target parasites, you would generally want to drive a T helper 2 response because you would release the antibodies that are antiparasitic, which can then stimulate eosinophils and neutrophils as well, but eosinophils to bind to the parasite. They sort of use the anti-parasite antibody, which is IgE, as like a little bridge, and it coats the parasite in IgE, and then the eosinophil comes along and says, ooh, I love IgE, and it binds to it and then degranulates and it kills the parasite. And so that these cells just kind of help with that. They stain with a basic dye. That's what the basophil comes from. I didn't say before, eosinophils, eosin is a stain, and it stains really like pinkish red, and the granules in eosinophils stain with that color, and so that's where their name comes from. Uh, Basophils, the opposite. So they stain like a deep blue, kind of cool, but they're almost impossible to find. They also do a little bit of antigen presentation. They can degranulate, but really something really interesting. So when I worked at the University of Pennsylvania, I went into a talk because we were doing some parasite work with schistosomiasis in mice. And I went to a talk from this guy. He was Irish and he had like the coolest accent. He was talking, and he was a PI who was, so that's like a boss, who was studying basophils. And he was revolutionizing how we understood basophils. And it turned out that everybody was studying the immune system the same way, where they would just like, they'd infect a mouse with like a parasite or whatever, and they'd look at it like, I don't know, 24 hours later, 48 hours later, maybe a week later, 2 weeks later. They would just look in the blood too. And they were missing where basophils were. So people were like, basophils are just this random thing we randomly find sometimes. But it turns out he studied basophils, and within the first 24 hours, they would traffic to the site of a parasite infection and then traffic to the nearest lymph node. And they were never really circulating in the circulature. And so that was really interesting. So people were missing how important basophils actually were for finding a parasite and presenting its antigens. And so that was really, really cool. All right, so moving on to natural killer cells. Okay, so natural killer cells are really interesting. So they're not myeloid-derived dendritic cells. They come from the lymphoid progenitors, and their job is to look at a cell, at your cells, and they look at your cells and they say, okay, do you have MHC class 1? So we haven't talked about antigen presentation and MHCs or HLAs. So in mammals, MHC stands for major histocompatibility complex 1. Every cell in your body, with like 2 exceptions, like red blood cells and I think sperm, with those exceptions, have MHC class 1 on their surface. So it's a really cool way of looking at a cell and saying, okay, cell, are you the right kind of cell? Are you supposed to be here or not? If you don't have MHC class 1, something is wrong. Okay, because every cell, for the most part, should have MHC class 1. So basically, you're either a cancer cell or you're a virally infected cell, and you also don't have MHC class 1, and you should also be killed. MHC class 1's job is to sort of act like a little glass bottom boat almost, and let cells, like T killer cells, look inside the cell. And so MHC class 1 will take all the internal antigens that are inside of a cell. So those are self-antigens. They could be a foreign antigen, like from a virus, and they'll put it on the surface, like they kind of hold on to it and show it to the rest of the body. And we're going to talk more about that next week. But if you don't have that, then that's really bad. And so it's kind of a check and balance. So a natural killer cell will say, will look at the cell and say like, okay, you don't have MHC class 1, so now you need to die. And so that's a good thing because it's getting rid of either cancer cells or virally infected cells or stress-killed cells as well. So I think I show a video as well right here of a natural killer cell binding to a cancer cell. So it's grabbing onto it, it's looking for MHC class 1, it's not finding it, and it's initiating apoptosis in that cell, which is different from necrosis, because if it's a cancer cell, you don't want to cause inflammation, which necrosis would cause inflammation. You want to cause apoptosis. You want to have that cell just kind of slowly break down. Moving on to macrophages. So macrophages, people have heard of. I apologize for the quality of this image. There's actually a few images in this slide set that are not up to the resolution quality that I typically like, but the pictures are just so good. I just couldn't resist. And so macrophages, macro meaning big, phage meaning to eat. They perform phagocytosis. They'll eat pathogens and chop them up and then present the antigens that those pathogens are made of. Their main job really is to eat a bunch of dead stuff. And so I think I have a picture here or a video of a macrophage like crawling around eating stuff. Macrophages serve a ton of purposes, and we're going to find out that's an understatement. So on the next slide, I'm going to show that. They have tons of different functions based on where they are. Macrophages also have 2 derivations. So they can either exist as a hematopoietic stem cell, they become myeloid progenitor, and then they become like a precursor, a monocyte. And monocytes are like circulating macrophages. And monocytes will then, they'll sense via damage or cytokines or chemokines, they'll sense a bacteria or something bad has happened. You get a cut or whatever, and it invades that tissue, and then it can start doing its thing. It can become a resident tissue. Now, there are resident macrophages that actually develop not from the hematopoietic stem system, or stem cell system, but from the stem cells for that specific tissue. So like in the liver, there are Kupffer cells that are derived from liver stem cells, not from the hematopoietic stem cells, which is really interesting. And so they're sort of reprogramming their stem cells to be macrophage-like, which is really, really cool. And so their main job, I just want to remind you, their main job is to eat bad guys and then present the antigen, because dendritic cells are sort of the opposite. Now, I have this slide here to show you just what some of these cells are. So in like every tissue has its own resident macrophage. And some of them have fun names, some don't. The liver is cool. Those have Kupffer cells, named after some dude named Kupffer. The brain has microglia, which is really, really cool. Those are fun. They serve a ton of different purposes besides just looking for bad guys. The skin has Langerhans cells. Those are really cool. What else are there? Most of the other ones are pretty normal names. Those are just the fun named ones. Oh, in the lungs, you have your alveolar macrophages. And those are the macrophages that will crawl around on the inside of your lung lumen and look for bad guys and eat them. That's pretty cool. Talking about macrophages, their partner in crime is the dendritic cell. Now, the dendritic cell, its job isn't necessarily to eat bad guys and chop them up, though it can do that. Its main job is to sort of show up kind of after all the damage is done and gobble up anything that's sitting around, which could be a bad guy, even though that bad guy may already be dead, and then present that antigen. So that's its main job, is antigen presentation. And they're called dendritic cell because they'll have these dendrite-looking things, kind of like a neuron has dendrites. So that's where their name comes from. And they really do kind of look like this. They have like little arms all over, which is pretty cool. Now, something we're going to encounter later on in this class and in the immune system in general, if you get deeper into this, the immune system changes a lot, especially as technologies improve. And it turns out, if you actually start looking at the RNA sequence data, so that's the RNA-seq up here, if you actually sequence what's going on inside these cells, so in here, these are our macrophages and our dendritic cells, and there's so many cells I'm not talking about in this class. There's just like a million of them. It turns out what this paper was talking about was trying to get us to maybe reclassify how we actually classify these cells and what their jobs actually are. Because you're trying to take all this historical information of, like old school stuff, like, oh, we know like this is a T helper cell. Well, now we have T helper 1 and T helper 2 cells. Oh, and then our technology improves and we're like, oh no, there's subsets of those. There's Treg cells, there's T helper 17 cells, there's T helper 9 cells. And so things get really complicated. And so, every 30 years you kind of have to reset. And that's what this is talking about. And the whole point of this is just to show you that things are really complicated in the immune system. All right, so we're almost done. And I wanted to talk about the last couple things. Are inflammation. So I realized that the quality of this picture is like the worst. I found it. I was like, oh my gosh, this talks about all the things I want to talk about with inflammation in like one picture. I was so excited about that, but the quality is just so bad. So I apologize. If you make it small on your computer, it's really, you won't even notice. But this is talking about inflammation. And I'm going to show, in just a second, I'm going to cut to the very end of that crash course video where he's going to talk about inflammation and fever. But I wanted to talk about this really quickly. So there are 5 signs of inflammation. Sometimes there's 4 listed. So that's redness, swelling, heat, and pain. Those are the 4 main signs of inflammation. If you have all 4 of those signs of inflammation happening in like your finger or whatever, you probably can't move your finger. And so lack of movement is generally considered the 5th sign of inflammation. And so how we actually generate inflammation is really, really interesting, especially where drugs come in and block those spots of inflammation. And different types of drugs, i.e. like Tylenol, all the other ones that block things, COX inhibitors, steroids, where they actually block the inflammatory process is really interesting. And that's something else. So if you go on to med school or something, that you'll actually learn like where we actually block those areas, which is pretty fun. And so this is just a really cool chart that shows just how inflammation can be resolved and generate homeostasis, or it can become chronic, which obviously is a bad idea. Now, going on a little bit more into fever. So the triggering of fever is really, really important. It's kind of a last resort to really, really trigger some crazy stuff to happen. In general, fever is a bad thing. Your immune system is still going to do its job without fever. But in general, bacteria tend not to be able to survive. They have a pretty narrow temperature range at which they can function. They might be able to live, but their temperature range for actually like reproduction might be pretty low. And so if you can raise your body temperature even a couple degrees, that could really affect a lot of the enzyme activity. And so that's generally why we cause fever, though fever is one of those, like it happens too often and it actually does more damage than good, as you may know. So give that kid Tylenol because his fever is 105 for 4 hours. And so it's not a good thing. But how you actually generate fever is really important and really interesting. And it's one of those weird connections between the immune system and the cytokines, or the proteins / intracellular hormones that we secrete, actually bridge the gap into the hypothalamus and control the temperature of the body, which is really, really interesting. So I want to cut to crash course, the very end of this initial crash course video, where he's going to talk about a couple of these things at the end.
So those are some ways your innate immune cells handle their enemies, but how do they know where to look in the first place? So let's talk strategery. So say you're in a banana factory, and you slip on a banana peel and you scrape your knees. Your outer fortress has been breached and the pathogens are just flooding in like orcs through Helm's Deep. Banana factories are very dirty places. Now your body wants to contain the spread of pathogens, clean up the mess, and get healing as quickly as possible, so it cues up your inflammatory response. This is basically an internal fire alarm, only it uses chemicals instead of sirens to get the message across, and instead of smoke and fire, you sense redness, swelling, heat, and pain. For example, in the event of injury, specialized mast cells in your connective tissue send out histamine molecules. And histamine is great at calling in the cavalry. For one thing, it causes vasodilation, which creates redness and heat at the site of the injury. Now, those things might freak you out a little, but they're actually signs of healing. The increased temperature, for example, ratchets up the cell's metabolic rate so they can repair themselves faster. Meanwhile, histamines and other inflammatory chemicals also increase the permeability of blood vessels, causing nearby capillaries to release protein-rich fluids. This causes swelling, which again is actually a good thing because that leaked protein helps clot blood and form scabs, while the lymphatic system sucks up and filters that extra fluid, cleaning it up before putting it back into your bloodstream. And of course, like chum to sharks, an inflamed knee is also going to attract a bunch of local phagocytes, which find it easier to escape your now leaky capillaries, and lymphocytes that are also flowing freely, helping to destroy pathogens while also cleaning up dead cell wreckage. And don't forget, during all of this, the neutrophils have been doing their best, but they were the first wave to arrive, so by this time, they're starting to die in heaps. They're triggered when the injured knee skin cells release chemicals that begin leukocytosis, the release of neutrophils from the bone marrow, where they're made, into the bloodstream. To attract the neutrophils to the damaged area, inflamed endothelial cells in the capillaries send out chemicals that act like homing devices. And when the neutrophils arrive, they cling to the capillary walls near the injury, flatten themselves out, and squeeze through the vessel walls to get to work. Your big monocytes eventually roll up to the battle and transform into hungry macrophages, replacing the first line of now dead neutrophils and basically just eating up any lingering enemies and then cleaning up the carnage. Now, all this works pretty well in most circumstances, but you may have noticed with a more major injury or an especially nasty virus or infection, sometimes your local troops get overrun. When white blood cells and macrophages run into more foreign invaders than they can handle, they let loose pyrogen chemicals that tap the hypothalamus and raise your body's thermostat, calling in a systemic fever to burn everything. The resulting temperature rise increases the metabolism of your cells so they can heal faster, and it also tells the liver and spleen to hold on to all of their iron and zinc so those things can't contribute to bacterial growth. But even then, sometimes you find yourself facing a more formidable foe. That's when you call in the specialists, your adaptive immune defenses. And to learn exactly how they save the day, you have to watch next time.
All right, so that's the end of that crash course video. We're going to see some more of those crash course videos because he's actually going to do a really good job and show some nice visuals, some good animations of how the immune system actually works. And the last thing I wanted to leave you with is that we're going to be talking about antigen presentation, and I promise I'll have a higher quality picture later. But the idea of taking a macrophage, having it eat a bad guy, chop up that bad guy, take those little chopped pieces of bad guy, and show that to a T helper cell, and show that to a B cell, and show that to a T killer cell, and say, hey, we should attack this specific antigen and kill it. And that's what we're going to talk about next time.